Hello, my name is Marty Sanders. I am a biology student here at the University of North Texas, and my project is the distribution of microplastic contamination in Denton, Texas stormwater retention sheds. So the Benthic Ecology Laboratory at the University of North Texas is currently conducting a large-scale study led by Dr. James Kennedy to understand how urbanization and watershed characteristics affect the population and diversity of benthic macroinvertebrates from 6 urban ponds in Denton, Texas. My portion of this study is looking at the distribution and concentration of microplastic contamination present in Denton, Texas urban ponds, as well as the effects or health hazards microplastics could pose to the benthic macroinvertebrates present in these ponds. So the samples for this project were collected at 3 of the sample ponds here in Denton, Texas. In this map, you can see here 3 of the ponds, North Lakes, Apogee, and South Lakes. For this project, I followed 2 methods that were described by Dr. Michael Claessens in the new techniques for detection of microplastic contamination in sediments and field collected organisms. Adult dragonflies were caught using aerial nets, and sediment samples were collected from each of the 3 sample ponds. The sediment and dragonflies underwent a nitric acid digestion, and then the remaining solution was then filtered over a vacuum filter. As you can see here in the photos, these are the remains that was left from the digestion process. All of these filters were inspected using a microscope. Apogee definitely had our highest concentration of microplastic particles found in the sediment samples. We also did find some microfibers present in the dragonflies as well. There was a little bit less at North Lakes. We did have some particles that we believe is styrofoam, and then a couple microfibers present in the dragonflies. And then similar results from South Lakes. We're finding the particles that look like styrofoam as well as microfibers. So for future research for this project, I'm going to be using a illustration column that was also described by Michael Claessens. This column separates particles based on density, size, and shape. So I'll be able to get the concentration of microplastics present in the sediment samples of all the ponds. I also will be using infrared or Raman spectroscopy for microplastic identification. Once this methodology is lined out, I will then move on to my second phase of my project, where I'll be looking at to determine the effects microplastics may or may not have on the growth and development of benthic macroinvertebrates present in these ponds. So I'd like to thank the University of North Texas McNair Scholars Program, as well as Dr. James Kennedy for giving me this opportunity. And thank you so much. Y'all have a nice day.